We're going to go over chapters 19 and 20 case studies, and the first case study is hemoglobin and oxygen transport. So we have Martha, who's a 27-year-old female. She presented to her local clinic with shortness of breath walking to the local shop. She experienced fatigue and lethargy. A blood test revealed that Martha was anemic. So explain how anemia can result in breathlessness upon walking to the local shop. So first we have to know what anemia is. So anemia can be one of two things. So first of all, anemia is basically just a decreased oxygen carrying capacity of the blood. So what that means is, if you remember our red blood cells, inside the red blood cells are these proteins called hemoglobin. And a hemoglobin molecule is a protein that's actually made up of four separate proteins. And so we call it a quaternary structure. And that means that it has, these four different tertiary structures, tertiary proteins in them. And at the center of each of those tertiary proteins is a heme group. And inside that heme group is a molecule of iron. So right inside there is a molecule of iron. In each one of those, there's an iron. And the iron has the ability to bind to oxygen. So each one of those binds to oxygen. So each one of these hemoglobin molecules can carry 4 oxygen. And the oxygen, and also the red blood cells have 250,000 about hemoglobin molecules. So really one red blood cell, if we multiply 250,000 times the 4 different oxygen that each hemoglobin can carry, it's really that one red blood cell can carry over 1 billion oxygen molecules. So one red blood cell carries a lot of oxygen. So anemia is one of two things. It could be a number of things that cause anemia. So possibly during anemia, you don't have as much oxygen or hemoglobin. So if you have a decreased hemoglobin, now you don't have as much oxygen carrying capacity. Another thing is that maybe you don't have enough red blood cells. So maybe the red blood cell count is down, and then you don't have enough hemoglobin to carry those oxygen. And then the other type is that possibly you don't have enough iron. And so if you don't have enough iron, and we call that iron deficiency anemia, then you also don't have enough oxygen carrying capacity. So there's also some other anemias that we have. There's like a pernicious anemia. Pernicious anemia is a decrease in vitamin B12, and vitamin B12 is necessary in order to create that red blood cell. There's also a sickle cell anemia. Sickle cell anemia is when the red blood cell is oddly shaped, and so it doesn't have the proper amount of oxygen in carrying capacity. It doesn't have enough hemoglobin, and so the oxygen carrying capacity is decreased. So all of this means, this anemia, what this all means is that you have decreased oxygen carrying capacity of the red blood cells, right? So you're not getting enough oxygen to the tissues. Well, oxygen is necessary. Oxygen is necessary to make ATP. So if you don't have enough oxygen, you're not going to be able to make ATP. And if you don't have ATP, the muscles can't function properly. And so, of course, when she's walking to the local shop, she's going to experience fatigue because her muscles are tired. They don't have the ATP that they need. In this second case study, we have Stu, a 32-year-old baker who decided to run a London marathon. He was very unfit and began his training 9 months before the event to get fit. On his first run, his heart rate was high. It was 190 beats per minute. His personal trainer explained that Stu's high heart rate was helping to increase his cardiac output. He also explained that with more training, the contractility of Stu's heart muscle would change to cope with the increased venous return, and his heart rate would not go so high after that. The first question asks, what is heart rate? Well, heart rate is the beats per minute. How many times does your heart beat per minute? What is stroke volume? Stroke volume is what is the volume of blood that's pushed out of the left ventricle during one heartbeat. So how are stroke volume and heart rate related to cardiac output? Cardiac output is how much blood is pushed out of the heart from the left ventricle per minute. So it's what is the stroke volume of an entire minute? And so we would multiply the heart rate times the stroke volume to get the cardiac output. So cardiac output is the heart rate, the beats per minute, times the stroke volume, which is the amount of blood pushed out in one beat. So now we're looking at how much blood is pushed out in 1 minute. So what is contractility? Contractility is the strength of contraction of the heart. And what effect will an increase in contractility have on ventricular pressure and the force of the heart muscle contraction? So this seems kind of obvious, but if you have an increase in contractility, that means the left ventricle is squeezing more, it's contracting harder. It's going to increase the ventricular pressure, and it's going to increase the force of the heart muscle contraction. Number 6, what is able to increase the contractility of the heart? Well, there's a number of things that could increase the contractility of the heart. There's the nervous system. So the sympathetic nervous system can increase the contractility of the heart, and those neurotransmitters, norepinephrine and epinephrine. And then there's hormones as well. So norepinephrine and epinephrine, when they're released from the adrenal medulla and they enter into the blood, then they're hormones, and they can increase the contractility of the heart. So can the thyroid hormone, and so can glucagon. So there's a number of things that can increase the contractility of the heart. What effect does increased contractility have on stroke volume? Well, the harder the heart beats, the harder the ventricle contracts, the greater the stroke volume will be. So how does it achieve the effect that you state? I think that question is just asking that if that left ventricle can squeeze harder, contract harder, then it's going to push out more blood. Number 8, name the 2 different pump systems that aid venous return to the heart during activities such as running. There is the respiratory pump and there's the muscular pump. So the respiratory pump it occurs when you're breathing, and every time the diaphragm contracts, it increases the volume within the thoracic cavity, and that sort of sucks that blood back up towards the thoracic cavity area, because fluid wants to move from an area of higher pressure to lower pressure. So when you increase that thoracic volume, you're decreasing the pressure. When you're making the volume of your thoracic cavity greater, that actually decreases the pressure that's in the thoracic cavity. So the pressure that's higher in the legs and in the arms, that blood then in the legs and arms is going to move towards the thoracic cavity where there's less pressure. The other one is the muscular pump. The muscular pump is just the muscular skeletal muscles contracting on those veins. So when the skeletal muscles contract and they squeeze those veins, it's squeezing that blood back up towards the heart, the thoracic cavity. The next question, number 10, is what is end diastolic volume? So look at the word end diastolic. So that means at the end of diastole. So that's at the end of rest. So we're looking at the volume of blood at the end of the ventricle relaxing. So at the end of diastole, the ventricle will have as much blood as it will have to pump out of the heart. So that's the end diastolic volume, which basically means How much blood does that heart have to pump out of that ventricle? And number 11, what is preload? Preload is the stretch on the ventricle wall as it fills. So the ventricle is filling up with blood and it's stretching the walls of the ventricle, and that is creating a little bit of tension on the ventricle wall, and that's what preload is. But Stu, once he started to strengthen his heart by exercising, so by exercising and training, he was able to increase the efficiency of his heart. His heart muscles actually would get stronger and the muscle fibers would get bigger. So if we look at, again, if you're looking at cardiac output, and cardiac output is heart rate times stroke volume, his heart rate was really high. It was 190 beats per minute in order to sustain the proper cardiac output. But later on, after training, he was able to increase his stroke volume because his heart was getting stronger. So in order to have the same cardiac output, he was able to increase his stroke volume, and that meant that he could decrease his heart rate to maybe 140 or less in order to get the same cardiac output. The next case study we have is the electrocardiogram trace. This case study is on Victoria, a 65-year-old female. She underwent a routine health screen. When she was younger, she was an athlete. She likes to stay very active, and ECG showed she had an increased PR interval. So the first question is, describe the normal pathway for conduction of depolarization through the heart. So we start at the cardiac pacemaker. So depolarization starts spontaneously at the SA node. The SA node is in the right atrium. It causes contraction of the atria. The atrial conducting fibers then carry that signal to the AV node, which is located in the intermuscular septum in between the atria and the ventricles. The AV node then conducts the signal to the AV bundle, which is located in the interventricular septum, and these fibers conduct the signal to the Purkinje fibers, which relay the signal to the muscles of the ventricle wall, causing depolarization and contraction. So question number 13, explain what an ECG measures and describe the normal waves that you would expect to see on an ECG trace. A normal ECG shows a recording of the electrical activity in the heart during the cardiac cycle. So changes in the electrical activity that's measured in millivolts are plotted against time, which is seconds. A normal trace shows a series of waves in the order of P wave, QRS complex, and a T wave. What do the main waves on a normal ECG represent? The P wave represents atrial depolarization, the QRS complex represents ventricular depolarization, and the T wave represents ventricular repolarization. Number 15, what does the PR interval represent and which parts of the conduction system does it test? Well, the PR interval represents the period between the onset of atrial depolarization, meaning the start of the P wave, and of ventricular depolarization, which is the first deflection of the QRS complex. The PR interval, therefore, represents the time for the conduction of signal from the atria to the ventricles, meaning how long is it taking the conduction of those action potentials to get from the atria to the ventricles. The wave, therefore, measures the time for the signal to pass from the SA node to the AV node, to the atrioventricular bundle, to the Purkinje fibers, and then to the muscular ventricular wall. Okay, question number 16. The problem in Victoria's heart is small in size and involves a single part of Victoria's conduction system. So which part of the conduction pathway do you think has been damaged in Victoria's heart? So the conduction of signal from the atria to the ventricles has to pass through the AV node, as this is the only available pathway to get from the atria to the ventricle. The AV node is a relatively small structure, and if it's damaged, it could increase the PR interval with no other ECG changes. Our next case study is on hypertension. So we have Pam, a 65-year-old female who's recently been diagnosed with hypertension or high blood pressure. Her blood pressure was 170 over 90 millimeters of mercury. She was prescribed a calcium channel blocker and an angiotensin converting enzyme, or an ACE inhibitor, in order to lower her blood pressure. So first of all, we need to explain what a blood pressure reading of 170 over 90 millimeters of mercury means. The 170 is the systolic pressure. So this is the blood pressure when the ventricle is contracting. And it's the blood pressure that we find in the arteries, in particular the brachial artery. The 90 is the diastolic pressure, so it's the amount of pressure in the arteries at the time that the ventricle is relaxing. What effect does the renin-angiotensin-aldosterone system, or the RAAS, have on blood pressure? And briefly explain how it does this. So basically, renin is released from the kidney. It causes the conversion of angiotensinogen to angiotensin 1. Angiotensin 1 then travels to the lungs, where the hormone ACE, which is the angiotensin converting enzyme, will convert the angiotensin 1 to angiotensin 2. Angiotensin 2 is a major blood vessel constrictor. So it's a major vasoconstrictor. And so that's going to increase the blood pressure. The renin also will target the adrenal cortex and cause an increase of aldosterone to be secreted. And aldosterone causes the kidneys then to reabsorb sodium and water. And so if you're reabsorbing more water, you're also increasing your blood volume, so you're increasing blood pressure. Number 19, how would inhibiting the production of angiotensin 1 help to reduce blood pressure? Okay, so the drugs used to do this are known as angiotensin converting enzyme inhibitors. So basically, if an ACE inhibitor is used, then angiotensin 1 cannot convert to angiotensin 2. And like I said, angiotensin 2 is a major vasoconstrictor. So an ACE inhibitor, which will not allow angiotensin 2 to be produced, will then inhibit vasoconstriction. And so that's going to reduce blood pressure. Number 20, what is a contractile cell and what is contractility? So the contractile cell is the cell in the heart that will contract versus the conducting cell, which conducts the electrical impulses. Number 21, what is meant by the term depolarization? Well, depolarization is when a cell becomes more positive. And so, in other words, when a cell becomes more stimulated or activated. Number 22, how do contractile cells in the heart depolarize, and what happens to the intracellular levels of calcium when they do? The conducting system of the heart is delivering action potentials to the contractile cells. So when an action potential reaches a contractile cell of the heart, it will open up sodium channels, and sodium will enter into the contractile cell. This depolarizes that contractile cell and causes the sarcoplasmic reticulum to release calcium into the intracellular fluid. Therefore, when the contractile cells in the heart depolarize, the intracellular levels of calcium increase. 23, how is calcium involved in the functioning of contractile cells? Well, when calcium is released from the sarcoplasmic reticulum, it binds to troponin. Troponin moves away tropomyosin from the active site, and that will allow myosin to attach to the active site on actin, and we get this cross bridge formation, and then we get a power stroke, and then we get a contraction. 24, based on the answers above, How would a calcium channel blocking drug help to reduce blood pressure? So let's review here. Here's our cardiac cell, and it depolarizes, so sodium enters into the cell. This is going to open up calcium channels in the cell membrane, and calcium enters into the cell. And so this is 20% of the calcium that the cell will receive. The calcium entering into the cell will cause the sarcoplasmic reticulum to release calcium, and this is the other 80% of the calcium that will be in that cell of the the contractile cell. So now we have 100% of the calcium that cell is going to get. The calcium then binds to troponin, and troponin moves tropomyosin away and exposes the active site, and then we get the myosin head binding to the actin, so we get cross-bridge formation, and then after that we get the power stroke, and this causes the sarcomere to shorten, so we end up with a muscle contraction. So the calcium is necessary for the contraction to occur. So if we block those calcium channels, then we're not going to have the 20% of the calcium entering into the cell, so then the sarcoplasmic reticulum isn't going to be releasing the other 80%, and the contraction or the contractility of the heart will be decreased. So we'll get decreased contractility. And we know that if we have decreased contractility, that means that the heart's not pumping out as much blood, and that's going to lead to a decrease in blood pressure. The last case study that we have is a calcium channel defect, and this kind of comes right off of the last case study. So Lee, a 41-year-old ship worker, fractured his ankle at work. The injury required surgical repair under general anesthetic. 1 hour after the operation, his heart rate rose, so he had tachycardia. His muscles went rigid, his body temperature rose, and it went pretty high. It went up to 104 degrees Fahrenheit. So his condition is a life-threatening condition. It's caused by a genetic defect that affects the calcium channels on the sarcoplasmic reticulum of the muscle cells, causing them to remain open. So question number 25 is, what is the role of the SR in muscle cells? Well, the sarcoplasmic reticulum is that intracellular organelle that stores calcium. And when it releases into the cytosol of the cell, it will start that muscle contraction by binding to troponin. Question number 26, when do the calcium channels on the SR normally open? Well, they're going to open when the muscle cell depolarizes and the calcium channels in the cell membrane open and calcium enters into the cell, and that opens up the sarcoplasmic reticulum calcium channels. What happens when the sarcoplasmic reticulum calcium channels open, and what effect does this have? So when the SR calcium channels open, calcium leaves the SR, enters into the cytoplasm of the contractile cell of the heart, and it binds to troponin, which moves tropomyosin away from the active site, so we get active site exposure, and then we get the cross-bridge formation where the myosin head binds to the active site, and then we get the power stroke where the myosin head will pivot, and that will shorten the sarcomere, and ultimately we get the muscle contraction. Question number 28. When do the calcium channels on the SR normally close? Well, as soon as the action potential stops, then the cell will stop depolarizing and the calcium channels will close. Number 29. What will happen if the calcium channels on the sarcoplasmic reticulum are open for too long? How does this help to explain Lee's muscle rigidity? So as long as the calcium channels are open and calcium is being released into the muscle cell will continue to contract. So if there's excess calcium, that means that those muscles are contracting excessively, and that would lead to muscle rigidity where the muscles are contracted for too long. Question number 30. How are calcium ions normally moved back into the SR? Is this process energy dependent or is it passive? So calcium ions are moved back into the sarcoplasmic reticulum after the action potential has stopped, and it takes ATP in order to activate the pumps that will pump the calcium back into the SR. This is a calcium ATPase pump, and it requires ATP to pump the calcium back into the sarcoplasmic reticulum. The last question, based on your answer to question 6, and given that the calcium channels on Lee's SR are being kept open, relatively how much energy will his muscle cells be consuming? And does this explain his body temperature? So because he has a problem with the calcium channels and the calcium's not being pumped back into the SR, so calcium is remaining in the muscle cell, and this means that the active sites are exposed and the contraction continues. Another thing that's necessary for muscle contractions is ATP. Remember that ATP controls the myosin head. It controls the myosin head from releasing and then recocking and then pivoting. So ATP has a big role in muscle contraction. So if the muscles are continually contracting because the active site is exposed, that means the muscle's going to need more ATP. So it's going to require a lot more energy in his muscle cells because there's increased calcium. And so every time ATP is produced, and ATP is a form of energy, we know that when cells produce energy, it will produce 40% of that energy in the form of ATP, and roughly the other 60% of the energy will be produced as heat. So this explains Lee's body temperature increasing to a dangerous level.